Hi, my name is Kate Levinson, and I'll be discussing the Levinson and Stahl DNA replication experiment. So, to understand the experiment, we have to understand that there are three different types of DNA replication. The first one is conservative, and it results in one molecule that consists of both original DNA strands and one molecule that consists of two new strands of DNA. Semi-conservative replication uses an old strand as a template to direct synthesis of new complementary strand and results in two DNA molecules with one original strand and one new strand. And the last one is dispersive, and it results in two DNA molecules that are mixtures or hybrids of parental and daughter DNA. So each strand is made up of original and new DNA, and the image to the right displays that. The experiment revolves around the growth of E. coli and the replication of DNA. So the two scientists began growing E. coli in nutrient broth that contained a heavy isotope of nitrogen called 15N. And as the bacteria grew, it took up a lot of the nitrogen from the medium and used it to synthesize new biological molecules such as DNA. After many generations of bacterial growth in the 15N medium, the bacteria was switched to a medium containing a light isotope of nitrogen called 14N. And the bacteria grew in this medium for many generations as well. And the pair took samples of the bacteria's DNA after rounds of replication in the different mediums and were labeled as 15N or 14N. Levinson and Stahl used a new technique called density gradient centrifugation to measure the density of the DNA. So they took their samples and were able to separate the two bands based off of the 15N and 14N content in the DNA. And the centrifugation allows little changes like that of 15N and 14N labeled DNA to be detected so they could figure out what type of replication occurred. The photo to the right shows the results of the experiment, and you can see that the observation prior to replication when it was in the 15N medium is just a heavy band because that's the original DNA. And then when you transfer it to the 14N medium, after one round of replication, there's an intermediate band, which was a mixture of the two DNAs. And then after the second round of replication in the 14N medium, there were two bands, one intermediate and one light. And then as replication occurred in the following stages, it continued to produce one intermediate band and one light band. So what does that all mean? The results of the experiment can be compared to the image above when determining what type of DNA replication E. coli is. So as we discussed in the last slide, after the first replication, the experiment produced a mixed band, which fits in with the semi-conservative replication and the dispersive replication. But then when we move into the second round of replication and the test with that, a light band and intermediate band were produced, which only fits in with the semi-conservative replication. So the conclusion is that DNA is semi-conservative.